everyone, and a warm welcome to Biotalk Series Week 4 on the topic Life and Death in Between Cancer Cells, conducted by the Department of Biotechnology, St. Joseph's College of Engineering, Chennai. Before we begin with the session, I have few instructions to be followed throughout the session. Please mute your microphones and do not introduce yourselves in the chat box. Post your questions in the chat box and it will be answered in the Q&A session. If you face any technical difficulties, post it in the chat box and we will resolve it as soon as possible. We will now start the session with Tamil Thai Valtu. I now call upon our event organizer, Dr. Justin Pakya Jacob, to deliver the welcome speech.
Good evening. Welcome you all for this Biotalk webinar series week 4. It is my pleasure to welcome our speaker of the day, Dr. Shiv Pujan from Sydney Kimmel Cancer Center, Thomas Jefferson University, US. It is evening 5 o'clock for us in India, but it is the early morning in US. Whenever I approach Dr. Shiv for this program, without any hesitation, he has accepted our invitation and joined this program for deliver his wonderful speech on this day. I welcome all the participants from Indian universities, various professors, HODs, and scientists, and research scholars in India, and also from various universities abroad. I also welcome all the teachers who has joined in this program from various schools in Tamil Nadu. With this few words, now may I invite Dr. G. Srikumar, Head of the Department of Biotechnology, to deliver the inaugural speech.
Welcome you all for another Biotalk lecture series, week 4, hosted by the Department of Biotechnology. I wish to brief some highlights about our college and the department. St. Joseph College of Engineering, one of the top colleges in India with the 160th position as per current NIRF ranking. It has got 26 years of high reputation in educational society with A+ grade accreditation given by NAC. It is having 10 UG programs and 7 PG programs with an affiliation from Anna University, Chennai. Our Department of Biotechnology is 18 years old with BTech and MTech programs. So far, we have bagged 139 university ranks with 10 golds and 7 silver medals and with 100% pass percentage every year. The department has got high-end laboratory facilities and fetched research center recognition to do PhD program and have more than 1.1 crores of rupees as government funding to conduct research. Despite this unforeseen situation, our biotechnology department still wishes to keep our people educated about the ongoing research and developments occurring in the field. This way, today's Biotalk lecture series is aimed on life and death in between cancer cell. Cancer arises from the transformation of normal cell into tumor cell. It is because of the interaction between person's genetic factor and three major external factors, that are physical carcinogens such as ultraviolet and ionizing radiation, chemical carcinogens. At least 66 known potential carcinogenic chemicals are disclosed, such as asbestos, aflatoxin, components of tobacco smoke, etc. We have biological carcinogens also, such as infections from certain viruses, bacteria, parasites, example human papilloma virus, Epstein-Barr virus, etc. Recently, the International Agency for Research on Cancer has categorized red meat, that is salted, smoked and cured beef, lamb or pork, etc., as high risk potential cancer causing agents. Cancer is the second leading cause of death, and around 9.6 million deaths happened in 2018. Globally, about 1 in 6 deaths is due to cancer reason only. The world of scientific community is fighting against the chronicles of cancer. However, as a patients who fight against it should understand that life is 10% what happened to them and 90% how do they react to that. In order to give more authentic research-based explanation about cancer, today we have Dr. Shiva Pujan, postdoctoral research fellow from Sidney Kimmel Cancer Institute, Philadelphia, USA. At this moment, I wish to appreciate the notable interest shown by the participants. There are 826 participants, including 173 faculty members and R&D personnel, 653 students and research scholars. The participants are from 11 different countries such as USA, China, Nepal, Philippines, Zambia, North Macedonia, Indonesia, Nigeria, South Korea, Pakistan, etc., and also from 22 Indian states. With this proud note, on behalf of the patrons, Dr. B. Babu Manohan, Chairman, Ms. Jessie Priya, Managing Director, Mr. Sashi Shekhar, Director, Dr. Seshagiri Rao, Principal, and Dr. Parvadavardhani, Dean of the college, I welcome the invited speaker and all participants to this Biotalk series. I take this opportunity to convey my warm felicitation to the organizing team, Dr. Justin Pakia Jacob, and student coordinators, and extend my best wishes for the success of this webinar session. All the best. Thank you. Over to Ms. Helen Benita, coordinator.
Thank you, sir. Our speaker for today's event is Dr. Shiv Pujan, and the topic of the talk is life and death in between cancer cells. Dr. Shiv Pujan is a prominent researcher who is currently working in Sidney Kimmel Cancer Center, USA, on a rare genetic disease, epidermolysis bullosa. He pursued his Bachelor of Science degree at CSJM University, India. He also completed his master's degree at CSJM University in the Department of Biochemistry. He completed his MPhil at Annamalai University, followed by postgraduate diploma at IGNOU and PhD at CSIR. Dr. Shiv Pujan is a proud recipient of various awards such as the Young Scientist Travel Award from the Department of Biotechnology and from the Department of Science and Technology, and Rambau Kulkarni Award for the Best Poster Presentation. He has research experiences in dermatology, cancer biology, epidermal stem cell, and environmental carcinogenesis. He has over 16 research papers published in renowned journals and presented about 8 papers at various international conferences. He has also conducted various training programs and workshops. We are most privileged to have you in the session, sir. I now carry over the session to Dr. Shiv Pujan.
So are you seeing my PPT?
Yes, sir.
Okay. Thank you, Arun. And thank you, Dr. G. Sekshi Kumar and Dr. Jacob. It's nice to hear lots of things. And yeah, first time we saw the videos, of the university. That's very nice. I think it seems a college and it looks like a university. It's very big. So today, as Dr. Sri Kumar already explained a little bit about the basic cancer, how the cancer is started and how the cancer is making lots of trouble. So I made this topic life and death in between cancer cells because the cancer cell somehow will behave like immortal cells and not a normal cells. It's become cancerous, and and the cells even not going for a death cycle and persist as a long life as a cancer cells. Before going for the further information about the cancer, so here in this presentation, I would like to just cover these topics. What is cancer and what are cancer risk factors? How does cancer spread? And can cancer be prevented? There is a diagnosis factors also. And what are the cancer stages? So, as we know, the cancer is the second most deadly group disease after the cardiovascular disease. And as here in America, 1 out of every 2 men in their lifetime get cancer, and 1 out of 3 women they get cancer in their lifetime. Is not a is very worst situation. And if you see in India also, I just get the data from 2018, around 1.7 lakh new cases reported every year. And out of that, 76% are males. Many patients came from treatment only after cancer has reached advanced stage, because in India, most of the cases not came early. Nobody knows when they get early stage of cancers. So the biggest problem in India, there is a diagnosis at early stage. So the cases reach to hospital, most of the third stage or fourth stage of cancer. As you know, the current list of cancer genes, which is germline mutation based cancer genes, are 70 genes. And the 342 genes are associated with the somatic mutation. So somatic mutation, normally when normal beings are able to normal life, there is no genetic problem, but they get any kind of influence from the environment, from the diet habit, or any kind of lifestyle change. These 342 genes are responsible for making these normal somatic cells to cancerous cells. And these 70 genes, as I'm working on these rare disease, that's epidermolysis bullosa, and especially these epidermolysis bullosa get cancer, it's very severe condition. So because of my interest is when these 70 genes get affected, they are not just affecting the one population, they are just affecting their progeny. If the one person get affected with any kind of damage in their genetic changes in germinal stage, they will carry their genetic mutation, and their progeny will suffer, because even they are not going to develop any cancer, but they get suffered because of this earlier mutation occur in their life. And I just one more very concerning things I found in the Indian paper in the Hindu published. Most of the range of female patients for the age group of 40 to 54 is higher than the male population. And it is very alarming situation for the female population in India, because these population is more vulnerable and they used to get cancer very easily. Or normally ovarian cancer cases are very prominent. That's why the 40 to 54 age group is showing higher number of cancer rate in female. So today's objectives I'm covering, and after ending this session, maybe you all understand what cancer is, state how a cancer diagnosis is made, identify cancer risk factor, and understand the importance of cancer stage. And there is a therapeutics, new therapeutics in techniques, what techniques are useful for cancer treatments. I also cover these. And also I discussed what is my part in this cancer research. So as Dr. Sri Kumar has explained a little bit about the cancer is not just one disease, it's a group of disease. And if we call group of disease means if you any cell of your body is not any foreign element changing your things or cellular homeostasis, it's in your cancer cells always in your body, but they are not active in form. That's how they maintain their status. For normally the cells is growing and their cells growing for the normal growth rate, and they died after their lifetime, their life period. And the cancer is abnormal growth because they disturb somehow because of lots of factors. Each cell contains a set of instructions. Basically, there's a genetic material that's the DNA. If certain things happen in DNA, even normally every DNA change in particular life in a particular population, we call this SNP, is a single nucleotide polymorphism. That's one kind of mutation, but that mutation is useful because somehow we change our identification from our other people. So and you see there is a certain changes in morphologically behavior. Every changes because of SNPs, but these mutation is not harmful. Sometimes this, if SNP also making some trouble, they also give you a mutation that's cause cancer. The instructions tells the cells when to reproduce and when to die. That's how the cell decide when they start reproducing their another cells and when they start to die, their life is completed. As every cell has their status for one cellular cycle to two cellular cycles or 100 or 1,000 cellular cycles. They already designed according to their telomerase. Sometimes these instructions get failed and the mutation occur. If cells keep reproducing in an uncontrolled way of mass, that's called tumor. But this tumor get worse when this get tumor metastasize. So as I choose this topic, the cancer cell traveling in between life and death, because as you see, normal cells start their life and there's an every checkpoint. They decide where to go. But the cancer cell just start growing and growing, not to stop. So there's a no stopping stage. So become immortal cells. That's immortal cells become cancerous. So if the cancer persist, there's a two possibilities. There's a normal cancer cells and it becomes senescent cells. Sometime these cancer cells reside in your body, but they behave like a normal, but because of some other stimuli, they will get excited and become a cancerous. So when I look into that, there is a story how the cancer get mutated when the normal cell cycle so is going on. So I just made one pictorial diagram to get more easier way to understand. For example, every cell has a normal life. So I just compare these cells as a car. So sometimes there's an abnormality. So the car which is going in the normal speed with a normal rate, there is no problem. All the brakes and oil is full and they are fine. They are not making any trouble. But somehow, if any disturbance occur in the way of the car, there is a proto-oncogenes which already in every cells. And these proto-oncogenes are not making any harm when they are inactive stage. Not inactive means they are not mutated or not converted into oncogene. So these proto-oncogenes, when they converted into oncogenes because of many factors, they leads to not able to stop that car, that cell, and these cells start moving fast without any control, and they become cancerous. And the thing is, when the genes are, you get the two copy of chromosomes. If the one copy is get mutated, the chances are, if you see most of the genetic disease, there's a one like female females are getting cancer from the parent, but not the male population get the cancer from their parents. That because of if the mutation occur in one set of chromosomes, and also the mutation not only happen once, it maybe has multiple time mutation, and it will cause multiple damage. Somehow cells is not able to repair those things. Just last month, a very big discovery published in a Nature journal, and they are describing how the cancer evolution occur, and how the cancer, when the cancer initiated, and how then the progression will start. So in the right side, there's a pictorial diagram. I just explain in a very easiest way. These cars get messed up and they piled up because of a big accident. So you don't know which car is responsible for this trouble. If you see closely, this red color car may be the first driver car which get disturbed, and because of this car, all other normal cars, they are coming in a normal way, they also get affected. So why I'm saying this picture to compare with the cancerous population, or if one cancer cell get affected by any mutation, or and that cells get abnormal, there's a high chance to their nick environment get affected. And if they get affected, every other normal cell also start behaving like a cancerous. So when the gray, you see the gray population, normal DNA replication is there, there's a normal organ development, but when any precursor lesion, any damage occur, there's a multiple time these lesions may maintain, and they will change their normal status to divide faster. Are they, if they capable to repair it, they will repair it. And there is also other mechanism. If they found something wrong, the other immunotic cells, they come to destroy these cells. But if all other things are stopped, then the primary tumor will generated, and this primary tumor is get invasive and become more heterogeneous tumor population. That's how you see different color here. So as I said, the tumor can be benign or malignant. So most of the cases, if you see, we get some kind of war kind of things in our body, these kind of tumor mass, but they are not the cancerous. We already know these just mass schemes. They will maintain the lifetime if there is no trouble. But if this benign tumor get any exposure, for example, if you have certain kind of tumor, and if you eat something bad, chemical carcinogens also there, environmental factors is also there. So lots of other factors. There's UV radiation. As I'm a dermatologist, so my work is basically based on skin. So if I say the UV radiation will cause the big trouble, and these normal benign tumor become malignant, so they start hyperplasia, dysplasia, the in situ cancer, and then invasive cancer. The invasive cancer get into all the bloodstream, and they travel all around the body, and they get migrated. And where they get migrated, they will generate their site tumor. So this is the final stage of the cancer. When you see the fourth stage, when the cancer is malignant, it's very hard to cure, because in that case, you cannot see where our treatment is going on. And the thing is, what causes cancer? There is a lots of things. Over time, a cell takes many hits, multiple hits. If a single hit there, the body cells has a very big and very powerful structure to maintain their normal homeostasis. But if they get hit multiple times, then somehow there is a single cell start mutation. As this is the picture I get the important paper that published in Nature. You see, they confirm not a single hit cause cancer. So they use a mice model. So just they give the carcinogen, and when they get the tumor, the gastric tumor in the mice, and they find after multiple exposure, cells have to maintain their genetic structure. But when the lesion is persist for a long time, there is a lots of mutations, like we have ATGC, so C2A mutation, C2G mutation, C2T mutation. So these mutations occur, and in India, most of the population get the C2T or T2A mutation because of Indian population. Most of the cancer cases based on the tobacco and other smoking. So the mutation, if the start from the driver genes, they start their downstream mechanism, and somehow the driver gene is a big culprit to make the cancer cell. So when we talk about the cancer risk factors, there are several factors, not only one factor. There are viruses infection. 10% virus infections participate in cancer, making the cancer cells. These kind of things are lifestyle based. Alcohol consumption, there's some unknown factors also. There are sexual practices also. But why the red area, you see the diet is a very important. They play in a globally, this is the global index data. If you see, diet has 30 to 35%, and also similarly tobacco has 30 to 32%. Is very big concern. In Indian population, maybe in India diet is very good, but this tobacco things is very common in India. Even this is common in rural areas also. That's why in rural areas, from last 10 years, I heard this cancer news exceeding day by day. So if we look the diet, if you manage diet balance, from this picture, you see this is the normal yin and yang from the Japanese mythological structure. They somehow say if you maintain your diet with some positive or negative, I say just from the meat part and from the green part, you just maintain a homeostasis in this diet chart, you will maintain your chance to get cancer from the diet. Their chance is less. But if you exceed any one of things, I'm not saying if you exceed just only green part and just only red part, they somehow disturb your diet, and this also make your cells normal to cancerous. And also there is a lots of things in tobacco. I'm not going to in detail because this is another big group of study. My concern is to just pay attention. There's some important also these occupational hazards. There's environmental pollution. Like in India, most of the cities in the world, they have higher rate of pollution. So somehow, if somebody is in very good diet and they are not using tobacco and they are very well and good and very healthy, but if their environment is not good, they will also not able to maintain their normal cell. In that case, their cell also get cancerous. That's how I'm saying if this cancer is not come from the one way, it's come multiple way. So it's everybody's responsibility to think how to make everything stabilized to maintain this cancer risk. And there is a other way, the family history. What as I'm talking about, if you get any genetic trouble, so there is a high chance to your offspring they will get cancer very easily. As you see in this picture, I put this zombies picture, as it is very common in America because they love to watch these zombies movies. So here, if one person become a zombies, they will try to attract any normal people. So if one normal cell get affected in a one family history, there's a high chance in their genetic pedigree they get in their second generation high chance of cancer risk. For example, if somebody is smoking very much, even they are not get cancer. But if someone is not smoking, but they're always nearby and there's kind of, they may be a passive smoker, but they get very low dose of that passive smoke and they get generated cancer. That because of in their families, somebody already get that cancer in earlier stage and their genes dominate in their offspring. So to continue with the risk factor, as you know, the family history and the exposed with the environmental and their occupationally viruses and radiations. And if you talk about the genetically, there is a oncogenes. When they get disturbed, they will start all the downstream application to stop the cellular homeostasis. And there is a tumor suppressor genes, P53 and very common APC. And there is a DNA repair genes like BRCA1 and BRCA2. And these are very common for the breast cancer. So I'm saying the cancer is a multiple genetic defect, not only one. And then how is cancer diagnosis made? First, when you get the symptoms, there is an early warning, non-healing oral or skin ulcer, lumps swelling in breast and neck, limb, dysphagia, hoarseness, hemoptysis, painless hematuria, postmenopausal bleeding, unexplained weight loss or anemia, change in mole size, color, and ulceration. And the diagnosis for early suspicion may be phenotypically, you see somebody has a lump of tumor mass, you can see something is wrong here. Then there's a radiological way, there is histological way to check the biopsy, and the most accurate way this time, the first way of identification is histological. When you see the histological picture, then you confirm there is a kind of tumor growth in a particular population, and there is a normal side area is not covered with the cancer cells. And there's a tumor marker. And these days, the markers they use for CA125 ovary, PSA analysis, CEA colon cancer, and there's a NSGT and so on. So when you see the primary cancer site this guy is an older guy. I just picked this picture because most of the cancer case in the United States is because of these 50 to 80-year population in this age group. So if you see in the liver area, if this cancer is localized here in liver, we call it liver cancer. If it is localized in the lung, is the lung cancer. And if it is localized in the brain, we call it brain tumor or brain cancer. Or in intestine or a colon area is the colon cancer. And also in a gastric cancer. So most of the Central Asian countries like China, Japan, and South Korea, they are very much affected by the gastric cancer. And if you look the histology and the morphology of the cancer in the liver, you see this, the white patches is a very metastatic tumor here, and their histological picture. And you see the lung cancer also is very much bulging with the tumor cells, is a yellowish structure showing lots of tumor cells in this lung. And also in the brain cells. Similarly, in the colon cancer, there are lots of nuclei with the tumor cells. And in the gastric cancer also. In these pictures showing these cancer, when they localize with particular reason, they are stomach cancer, but in this stage, they are malignant. So type of cancer treatment like surgery, chemotherapy, radiation therapy, they are very well known and very, very much used from for last years. And the targeted therapy and the immunotherapy, they are now in a very progressive way. There is a stem cell or a bone marrow transplantation also, and there is a hormone therapy. These are green signals here. The targeted therapy I just pointed out because recently these therapies are much progressive. So I'm going to talk much more about in this targeted therapy or immunotherapy. So there's a new technologies for cancer treatment from last year to last year. I just get from global data. These days the CAR T cell population, the CAR T cells technology is very popular, and they are going to be preclinical to phase 1, phase 2, or phase 3 trial. Others also, the NK cell and NKT cell, novel T cell or technology, TAA, all these cells basically are immunological therapy. And these therapy giving some hope in near future. If we get very close to get any molecule, we are successfully make these CAR T cells, definitely there's a big chance to get these cells in early stage, even in the later stage, both way. If you see the trends in cancer therapy pipelines, the year by year, if you see the solid tumor, if you treat a solid tumor, there is a high chance all other possibilities, the TIL cell or TCR cell, all these therapies are applicable. If you see in 2019 or 2020, all therapies are applicable in solid tumor. But if you look here, in 2020, 2019, you see there's a CAR T cell population is much higher because these CAR T cell technology is way better than the other. And their targeted based therapy is also very applicable. And the top 10 countries, those countries are investing a very much fund on to develop these technologies. United States is one of the biggest. And they, till now, there's immunological, oncological agents around 1,837 molecules till now they identified, and out of that, there's a 295 active molecule. They are now used in America. Even China is also using lots of clinical trials and lots of molecule identification. And if you see the top 10 countries, the Netherlands, Canada, Switzerland, France, South Korea, Japan, and Germany, and UK also, they all are doing well. But if you see the graph, US is doing much hard in this T-cell therapies. So if you see the overall overview of 3,876 active molecules, and these molecules, the green saying only 620 approved till now, and they are now used for treatment of cancer and different cancers. And if you see the immuno-oncology global drug development, And if you see the graph here, the pie chart showing most of the CAR T cell technologies, China is using a huge number. India is doing also in one location, but is not in a big number. And other countries as well. But in both two countries here, China and USA, they are focusing more on this immuno-oncological technology. So now I come to my research path where I started. So I started from CSIR. I used to develop engineered epidermis in the lab. So I started in the mouse and also confirmed in the human population. I just take the skin sample from mouse as well as from human sample, and then get isolated epidermal stem cells. And from these epidermal stem cells, I developed the method how to regenerate these stem cells to become a skin cells, to become a skin, complete full skin. And that skin may be useful for grafting also. But my purpose to understand the cancer population, so I checked arsenic toxicity, because my project is based on the arsenic toxicity. So I used to use this arsenic, and I get lots of research paper based on arsenic toxicity, and this arsenic causes cancer. And this arsenic causes cancer not only in a single stage, that cancer may be in a genetic way. I will discuss in more detail. And that work, when I established this work, I published in a good journals. And then I moved to South Korea, where I continue to identify these cancer cell population. Here, I developed the new method to change the cancer cell to normal cell with the help of reprogramming factor. As you know, reprogramming factors, Japanese scientist Yamanaka discovered if you put these 4 genes in any normal cell, so any normal cell become a pluripotent stem cells. So my idea is if we use these 4 factors in a cancer cell, what will be the changes in cancer cells? So I just get publication in this work also. In this picture, if you see, there's lots of heterogeneous population, and when we put the reprogramming factors, these one population is become normals, and they are more homogeneous. So this homogeneity is also a big change in the cancer development, basically in the recurrent of cancer. When you treat some cancer with a chemotherapeutic drug, but somehow you see in lots of cases relapses because of cancer drug resistance. So my idea in this work to find out these resistance cell population and how to change this resistance cell population to become a normal and get this drug, and they will die normally. And then here, last year I published one paper which is also related to cancer, but this is more related to so arsenica is a kind of chronic exposed metal that cause cancer in a longer time. But the concern is when most of the farmers they are working in whole day in the land, if arsenic is there in their body and they get exposed by the UV radiation, then the chances is very higher to get cancer in their body. So I used to give arsenic transplacentally, and this transplacentally purpose is to understand if I give arsenic to mother, what are the chances to their litters or their pups get exposed by any changes in their genetic material, or their stem cells are in normal stage. So for this reason, I expose these mother with the arsenic diet, and then we isolated skin from the pups, and then isolated stem cells from the epidermis, and checked these toxicities much higher. And when you looked at two doses, because I use one lower dose and one higher dose of arsenic, one lower dose is minimal, because if WHO recommendation is 10 ppm, I just use 10 ppm to 42 ppm. And you see a big number of changes here. Is a control. First one is a control. Second, green here is a low dose and is a higher dose. So definitely higher dose will change lots of in their genetic material. But low dose also affect the cancer cell population. This is the normal cell population. So for this, I just try to confirm this experiment from in vitro to ex vivo. So we get the cells, these transformed cells, and put into the nude mice. And when we see after putting these cells into nude mice, there is a small lump of tumor mass is developed. Is a positive control here. First one and second one is a arsenic exposed cells. I introduce these cells into this nude mice, and this is a control one. So this is the first important things we get from this arsenic. This exposure give in the counterpart population, in the F1 generation, to generate cancer. There are high chance to generate cancer in this population. And the last one, I want to make something to eradicate or somehow reduce this population, cancer cell population. So I use two nutraceuticals. These very common, as you know, the curcumin and another selenium. I use these two combination combinatorial therapy. Same way I just start giving the diet of these two nutraceuticals at the same time when I give the arsenic, and did the similar thing. And after whole experiment, I found they reduce this arsenic burden. And then what I did in South Korea, I exposed the lung cancer cell population. These lung cancer cell population exposed with reprogramming factors, and this reprogramming factor changed these cancerous cell to another kind of cells. Even still we are working on it to identify what kind of cells is, but their behavior is completely changed from their lung cancer cell population. Even they are not coming from same marker is not coming in their lung cancer cell population where they reside. So in this experiment we confirm these cancer cell get a transition. For example, if the cancer cell get drug and this chemotherapy induces and they started killing most of the cells, but few cells they somehow they resist these drug and still maintain their small population in the patient cells. And somehow, if any stimuli come again, or when we stop the drug treatment, these cancer small cell population relapsed, and they will come back with a big bad cancer cells, even much bad from the previous one. So somehow from this experiment, we confirm there's a transition in between 13 to 15 days. These cell population get matured and catch cancer. So what I did, we collected one of single cells, and each single cells I get sequence. So this is the sequencing data from 10x genomics to get one single cell has changed any genetic behavior or any morphological changes. We confirm this thing after we get this data. As population is migrating, these are one population, red to orange to green, and this last one is the heterogeneous, is showing this. The orange is the transition where cells maintain from 13 to 15 days, and between in this stage, if we plant our drug management, in this case, we can easily manage to overcome this reoccurrence of cancer cell population. And in the last, here I'm working on epidermolysis bullosa. This is a very rare disease. Like you heard about the xeroderma pigmentosa, this is also a very rare disease. And in this disease, in epidermis skin, we have epidermis and dermis. So in the epidermis and dermis, we have collagen to attach epidermis to dermis. If we lose collagen 7, 1A1, somehow disturb the dermis to attach epidermis. That is the major cause of this genetic disease. And this genetic disease not happen after any other exposure. This is came from their genetic pedigree. So in that case, if this skin is not attached with dermis, it start making blisters and is really very painful. If you see, look, is their chest. It is in chest, but some patient has in hand, there's toes everywhere. In lips also is very painful. And because of genetic disease, this is started from the birth. That's how it's very severe. And I'm working on if these EB cells, these epidermolysis bullosa, get any exposure. For example, if you get a scar and this scar get exposure of UV radiation, their chances are very high to get squamous cell carcinoma. And this is the stage when this disease get very worse. And my main work is to find out the molecule. So I am right now screening FDA approved drugs, or more than 2,000 drugs, to find out one single drug to somehow reduce this growth of this purple cell population. These are squamous cell population in epidermolysis bullosa. So we find out enzyme which is very responsible for making this squamous cell from this particular disease, and we are looking to somehow inhibit this enzyme, and we are working to make a transgenic mice. We are working on designing a mice which has this gene in increased amount, so we can easily understand if we put some drug, what will be the changes in their genetic environment. So last, here's the conclusive summary. The cancer is not disease as we confirm it, is a homeostasis disturbed by cells. Successfully, I attempted the pure population of epidermal stem cell, that what I did in my PhD. Changes the gene expression profile, which related to the tumor initiation. And the prevention also, I did these combinatorial drug to prevent these effect. And the recurrent cancer will change after the reprogramming of cancer cell population. So that's, and this I want to acknowledge these people. They helped me from my PhD to my here senior postdoc. Thank you. And the moral, the final word I put it here: mortal is at ease, and if immortal is a disease. Thank you. Questions?
Thank you, sir, for the informative session on the disease cancer, their evolution from proto-oncogene to oncogene, their progression, the cause of risk factors, the primary cancer sites, and the new therapeutic techniques. We also came to know that cancer is caused by multiple defects and not only one. I also thank you for sharing your valuable research on cancer. Thank you, sir. We will now move on to the Q&A session. I'll read out the first question to you. The first question is, how successful is a nanotechnology-based cancer treatment at present? Can it be a great replacement to chemo or surgical treatment?
Yeah, so these days, lots of nanoparticles synthesized. Basically, if I talk about the curcumin, say there's the titanium oxide, or lots of nanoparticle based small molecule, they are giving these drug to some in some stage. But right now, if I say practically, all the molecule, if you say the nanotechnology is just a very nano form of any molecule. If you say if the molecule itself is in a very nano, if you give, I'm just, I'm not working on nanotechnology field, so I just say if you put any molecule, it's already in a nano phase, and if you give this to any patient, they show some kind of changes. So these days, already these nanotechnology based, some are very good. There were some, for I heard some synthesized impregnated nanoparticle like titanium dioxide. They are doing very well, but yet not proved. So I cannot say is exactly what they changed. The chemotherapy are much better therapy than the previous one. I'm not sure about it, but definitely these days most of the concern is immunotherapy. So I'm hoping definitely this nanotechnology also gives some hope to treat these cancer cells.
Thank you, sir. The next question is how the environmental factors in epigenetical studies gives information of one place related to cancer possibility in people those who are living in another place.
Can I get in? Just give me some more clear. I just missed the point. The, can you say
Okay,
it again?
I'll read it to you again.
Yeah, thank you.
How the environmental factors in epigenetical studies gives information of one place related to cancer possibility in people those who are living in another place.
Right. I understand. So, for this, if you say one example, I am telling you, for in South Korea or Japan, these countries are very much affected by the gastric cancer gastric cancer, because in before 30 years back, these countries are severely affected by the food habits. Because of that, they are now suffering from this disease, this gastric cancer, basically gastric cancer. So I suggest this because of these environmental effect at that time will still they are facing this trouble epigenetically. That's how these epigenetic changes somehow persist there, and they are getting their situation to become a we can say this environmental factors also. They are affecting this because of this. So if you apply the same statics in India, we cannot say the same kind of environmental factor is making gastric cancer in India. Maybe some other factors here. So that's why these days most of the microbiome research is going on. They are checking the particular environment. For example, if you are living in Bangalore or somebody living in Kashmir, the both two places are climatically different. But if their epigenetic changes completely varies because of their environmental climate or environmental factors. So this is based on the place where they started and the environment.
Thank you, sir. The next question, can we add the antioxidants directly into the cancer cells to know its anti cancer activity?
So if you put any herbal molecule in a cancer cell, definitely they will show some activity. But this is not the cure. The thing is, the cancer cell is somehow stopped. We need to follow the death cycle. That's the main goal. If you just try to use any nutraceuticals, they will definitely give some rest, some kind of effect to stop at the cell cycle divisional stage. Maybe they will arrest in G2M phase, but they will not maintain this phase for long term. If you stop adding, for example, if you take curcumin or any herbal things every day in your life, may somehow you maintain your life. But if you, in the same time, you start eating the bad food or bad any you exposed by the chemical, you exposed by the UV, definitely when you stop eating these nutraceuticals, you will get the cancer. That's how they somehow maintain it, but these kind of nutraceuticals is not the, they can preventive medicine, but they are not the medicine which can treat the cancer. So you can use these nutraceuticals or antioxidants with any other chemical compounds or the any other molecules which are targeting those cells to go to decide these cells for the death pathway or the death cycle. That's how it's useful.
Thank you, sir. And the next question is, P glycoprotein receptor modifications makes cancer resistant to chemotherapeutic drugs. Is it possible to use virus vector as drug carrier to overcome this?
Yeah, these days lots of work is going on this virus based therapy also. But as you know, you are putting one genetic information with another genetic information. Somehow it is useful, but these inactive virus particle, they change. Yeah, definitely they're useful, but how long they will maintain, or every time like a vaccine, if these are if vaccine will develop, definitely this is very promising field. So is a possibility, but as the possibility is when the cell is in early stage or is homologous clone. For example, if you have heterozygotic clone, lots of heterogeneity in the cell population, you designed one receptor in a particular cell surface glycoprotein, but another which cells is this heterozygous from the previous one, then how can you design the same? So at the early stage is very applicable, but in the later stage is very hard to predict to reduce this heterogeneity. So that's how what I plan. I make the cancerous cell to homogeneous population for a certain period. For example, if we put it for 13 to 15 days. So in between these 13 to 15 days, if somebody use this kind of vaccine based or any virus based therapy, somehow then we can track 99% that population. That's how we can treat it.
Thank you, sir. The next question is, how do we collect the cellular DNA without getting affected by toxic chemicals for DNA electrophoresis?
If you want to isolate DNA, there is a way you are what DNA you are going to isolate. If you isolate genomic DNA, you from the cells. So what effect is going on in the cell is already in the cytoplasm or the in extracellular matrix, the ECM. And if you go the tissue level, if in their SNC environment. But if you get the nucleus, this this nucleus has a genetic material DNA, and this DNA, if you isolate this DNA, this already get affected by any kind of stimuli. There are signals already going on, so you cannot say any kind of adduct is there. That signal is already started. You just get the genomic DNA from normal phenol based method. You can just isolate the DNA, but then you can check what is the changes in DNA. You just sequence your DNA and see there is a mutation, what kind of mutation is there, how many mutations is there. So there is a study. First you get the DNA, whole genomic DNA, and you get that DNA, you find out the mutation.
Thank you, sir.